şey tamam.
No, but like you're dressed up, so I was wondering if you're.
Consul General of Turkey.
Oh, that's.
Ben lock edeceğim. Şey girdikten sonra Jack Shostak komiteyi hem lock edeceğim. Hi. Hi, Joy. Ah,
Time for the committee.
Jack is here. Jack is here.
Is here.
Yeah, I'll make him co-host. I mean, since he's here, I can lock it basically anytime.
Yeah, it's.
So I'll just make him the co-host.
Mistake if you log Jack out.
Yes. I mean, in the like right now they just cannot text or like cannot talk, so it's all fine.
Can he hear us?
Yes, he
Hey,
can
Jack.
hear. He can hear, but we can't hear him actually.
Oh,
Like he can hear, I think. But we will get that done once we're like,
yeah.
yeah.
Okay. Excellent.
But Jack is a co-host now. Yeah, so if you actually talk with this, it'll be fun, and then I can get this one.
Okay. So you don't want me to use the big one?
Oh, that's just for the like Zoom.
Oh, that's for the Zoom. Yeah. Okay, good.
I'll get this after you're done.
Sure. Well, we have time.
Mm-hmm.
I always wait a little bit.
Yeah, let's just wait
because some people have think there is actually another thesis.
Yeah.
So some people were going to the 2 to 3 thesis. So they'll start showing up in a minute.
Yes.
So
Pages here.
excellent.
You're done. I mean, Jack is here, so
Yeah, no, it's
most family
yeah.
friends are here.
So he's right. Just lock it, and then it won't be this annoying admit stuff.
Mm.
Very nice.
Breakfast Foundation.
Oh yeah. This guy's great. Very nice.
I'm just waiting for the comedy.
Yeah.
First.
Yeah, I suspect the other defense kind of was at least one of them because it was somewhat related. We had some attempt at observing last time.
It was cloudy, right?
Yeah. It was, well, it was not badly cloudy, but it was, the moon was full, so it always creates this scattering environment. So you can't really see faint things like nebulae, galaxies, because they have low surface brightness. So those additional photons really float.
Yeah.
We call it bright time. People who study galaxies actually don't even use the telescopes during full moon.
Mm-hmm.
They can't because the low surface brightness kills them. So the chair is here. Oh, so you do get some still. See, they do appear on the bottom, on top. Yeah. Let's see. Let me see.
Thanks for coming.
Yes, no problem, no problem. Yes, yeah.
So
Okay.
the protocol is I'll introduce
Yes,
him,
exactly.
and then when he finishes, we talk, or what do we do?
Exactly, exactly. So when if he finishes questions, and then then everyone steps out, and then we discuss. Yeah. Yeah.
Also, like after it, I can just close Zoom and you can call Jack from beyond. Right? That's also fine.
Yeah,
If you're
It's
coming
hard for people to
Jack,
leave.
Yes,
Jack,
yes.
Jack doesn't need to see anything.
Right,
Just
but
need to
after
talk to him.
Exactly.
Yeah.
After
So
it,
what
you can
I'm
just
going
call
to
him.
do is I'm going to call him on his cell
Yes.
phone.
Okay, let's
Exactly.
do it.
Perfect.
Yeah.
Then I will just turn off Zoom and close
Yeah,
it after
yeah,
it's done.
Yeah, sounds good, sounds good.
I'll
Okay,
just tell him or text him
Yeah.
so he knows.
okay. So I have to go to the airport.
At what time? Oh, okay. Let's do it. We don't need much time. I don't think so.
And just waiting for Marcus.
No, not necessarily.
Marcus?
Greiner.
He's a committee member?
Yeah.
I mean, he cannot join in person for sure. He's sick.
Oh, I didn't know he got sick.
Yes, yes.
Is he okay?
I mean, was he supposed to join?
Yes.
But I mean, we didn't
That's why I
hear
was
back
waiting.
from him recently.
That's why I was waiting.
Then we can start.
But if he's sick, I was going to start anyway.
I'll just lock the Zoom and then we can start.
Okay.
We just lock the Zoom.
Let's get the party going.
Hold on, let me just lock it.
No, don't worry. I'm just going to make fun of you right now, so you don't have to lock anything. Okay.
Guys, every time somebody joins, there's a ding ding.
At Harvard here, we have the 7-minute rule, which is when something says it's going to happen at 3, it's actually 3:07. So I'm starting a bit early, 30 seconds early. Oh, I'm too old. I don't read memos anymore. Is it the new president, the old president who canceled it? Oh, they made it a 15-minute. Yeah. Okay. Okay, well, They can't fire me. I'm tenured, so it's okay. Okay, so first of all, welcome everybody to Furkan's PhD thesis defense. Thank you for coming. I want to introduce the proceedings, and I will start. This is a PhD thesis, and every good PhD thesis project starts with a puzzle. And in this case, what is the puzzle? The puzzle is the following. How come we are doing a PhD defense in the physics department when the topic is about biology and the thesis is done by a graduate student who works in the chemistry lab of an astronomer? Anybody venture to? It's a one-word answer. The one-word answer is spin. Spin. Now, to explain that one-word answer, we have the best person to do this here, which is Furkan. And I want to introduce Furkan. Furkan comes from Turkey. He was born on the shores of the Black Sea, Trabzon. And he went to college, to university in Bilkent University in Ankara, Turkey, which is a new, very modern university with a lot of great equipment. He finished his Bachelor of Science there in 2018. He did a number of actually very exciting summer undergraduate internships around the world, including Indiana and ETH, and then Harvard here, where he became a graduate student, not surprising because he did extremely well, and joined the lab of Markus Greiner, one of our colleagues who unfortunately got sick and cannot be here today, who is an expert in atomic and molecular fundamentals of physics. And that's how Furkan got his master's degree in 2021, when he decided that the mysteries of solid state physics are not enough, and he decided to solve the mysteries of the origin of life. So he joined my group and my photochemistry lab in 2021. And now you will hear about what he did in those last 3 years. What he did is quite amazing, and he is quite an amazing student to begin with. Where do you get a student who did just in 2 years 7 papers, 5 of them as first author, 1 patent related to what you're going to hear. And on top of that, he's a blue belt in Jiu-Jitsu martial arts. Members of the PhD committee, take note of that. And he has a YouTube channel. He's an influencer. He actually helps other people, which has more followers and views than Truth Social, but doesn't make any money. There are similarities there. And he does so many other things. And yet, he's managed to contribute something very fundamental, not just to biology and periodic chemistry, but as you will see, it all has roots in physics. And that's why we're in this building. So Furkan, it's a great trip, but we want to hear the puzzle, the answer to the puzzle. Why spin?
Thank you so much. I'll get the microphone as well.
You did? No.
Well, thanks so much for the introduction. It can only go downhill from here. So no pressure, I guess. Yes, so first of all, welcome everyone who is here and making the effort to come here and listen to me. I am really, really honored for everyone's presence here. So today I'll talk about the chirality problem as it pertains to the origin of life. And we have to click on the screen, I guess. Perfect. So I will start with the origins of life and the homochirality problem and what it is. And then I'll talk about the interaction that we use to attack this problem and then the experimental results and how these results fit into an environmentally plausible scenario. And then I'll talk about how chirality established in one compound can spread out to the rest of the chemistry. So what do we mean by the origin of life? We kind of mean a chemical beginnings of biochemistry in a completely abiotic way from essentially, we can think of it as the transition of geochemistry to biochemistry. And of course, this is a big problem, and it has multiple stages. It starts with an environmental feedstocks that accumulate in a lake environment, like kind of the warm little pond as Darwin envisioned. And then these feedstocks with some energy that can come from sunlight and some other sources, basically through some chemical processes first give rise to monomers, biologically important monomers like sugars, amino acids, and fatty acids, and nucleotides. And then the second stage is these monomers are, of course, important, but they are by themselves not functional. And the second stage is that they kind of get activated and polymerize into functional polymers, which can actually do genetic functionality or enzymatic properties, such as RNA, peptides, and vesicles. And after this chemical stage, the first like self-sustaining protocells, the first ever cell-like entities basically start roughly around 4 billion years ago. And then after long years of chemical evolution, thanks to the functional RNA that was synthesized in these environments, we can get to a stage of populations that are known as last universal common ancestor. And then after, again, long evolution, we basically go to living entities that are called stromatolites, which we can actually nowadays find as fossils, for instance, in Western Australia. So this is the general overview of the origin of life. And it's very important that the geochemical feedstocks can essentially synthesize biologically important monomers through prebiotic chemistry. And now I'll talk about like what kind of prebiotic chemistry we are talking about and what it is. Yeah, you may have heard of like Miller-Urey experiment, which is a seminal experiment which was done in 1953, which was using a gas chamber and some lightnings, and it would synthesize lots of amino acids, but in trace amounts in various other compounds. So their further utilization in functional systems is hindered. But recently, a new system of operations, essentially, of doing chemistry was developed by someone who is actually sitting in the audience, by John Sutherland, and his presence really honors me. And essentially, him and his group basically discovered a very high-yielding and selective chemistry, which can synthesize shared precursors of life, including nucleotides, amino acids, and fatty acids from a very simple molecule called hydrogen cyanide using UV light and electrons ejected through the energy that is coming from UV light. And that is called cyanosulfidic chemistry, which essentially makes biologically relevant molecules in very high yields, but also it doesn't make the molecules we don't want. So it is a very highly developed chemistry that is actually seminal for our understanding in the origin of life. And if that wasn't enough, it is very interesting that the amino acids that John's chemistry makes are precisely the ones that correspond to the most nucleotide sequences in the genetic code, which probably are the earliest amino acids that emerge. The genetic code, as you know, basically is the alphabet that converts certain nucleotide or the anticodon sequences to amino acids. And the ones in the so-called family boxes are the amino acids that are synthesized by John's chemistry. And if this is not really interesting, I don't know what is. So essentially now our work will be related to this system, but it will address an important property that was not solved by John's chemistry. And that is the chirality problem. So in cyanosulfidic chemistry is an amazing paradigm, but it makes molecules in a so-called racemic state. And I will explain what that is. But before, I'll talk about chirality. So chirality is a geometrical property, and objects are said to be chiral if their mirror image are not superimposable on each other, just like our hands. And biologically important molecules are also chiral. They exist in left and right-handed forms in equal amounts if they're synthesized from a purely chemical pathway. Amino acids, sugars, nucleic acids are all chiral, and they can exist in mirror-symmetrical pairs as left-handed and right-handed mixtures. But when we look at biology now, biology is extremely picky when it comes to chirality. It selectively uses left-handed amino acids and right-handed sugars and nucleic acids. So essentially, biology breaks a symmetry which doesn't exist in chemistry. So how are we going to address this problem? And moreover, this is a feature of life, but it's also an important feature of life, because if we kind of think about the origin of life, by some way or another, we need to think about the genetic code that can basically transmit information and replicate, right? And do some maybe catalytic activities, and the sequences that show important catalysis properties need to be replicated as well. So self-replicating RNA, like RNA in general as a genetic code, is essential for our understanding of life. And turns out that RNA is actually a structure that is composed of chiral units, and its replication is true by base pairing, by so-called Watson-Crick base pairing. And because chirality is a geometric property, if the helicity of the helix is basically wrong, or is a mixture of left-handed and right-handed mixtures, it is impossible to find a match for the strands with each other. So it is impossible, basically, to replicate RNA if it is composed of heterochiral nucleotides. And this has actually been demonstrated experimentally some time ago. And essentially, we can say that without figuring out homochirality problem, we cannot understand how functional genetic code emerges, and without that, we cannot understand the emergence of life. So essentially, if we go back to the diagram that I showed before, the transition of geochemistry to biochemistry, in the stage where we make biomonomers in a racemic state, we need to figure out the chirality problem such that the polymers can be arranged in an orderly state. And if the strands are homochiral, then their functionality will be satisfied, and we can have self-replicating functional RNAs. So essentially, homochirality problem sits at the very center of our understanding of life's origins, and without which we cannot really create protocells in the lab. And because of that, homochirality problem was recognized by Science magazine as one of the biggest questions in all sciences. In fact, it is listed together with origins of life as a separate problem. And the question is, we don't know what triggered this symmetry breaking, and what is the physical reason behind this feature of life on Earth. And my talk will be about this massive problem. So now I'll break the problem in parts, because that's what we do when we face with a hard problem. So we start from a racemic mixture, and that's what you get. If you do a chemical synthesis, you get an equal amount of each chiral molecule. And then if there's an agent that breaks the chiral symmetry, or couples the chiral symmetry strongly, and introduces a chiral bias, or an enantiomeric excess, as we call, then we kind of introduce basically symmetry breaking utilizing that agent. And then that excess can be amplified to homochirality. But that is not enough, because life is not a single molecule thing. It requires many molecules. So making one compound homochiral is great, but it's not sufficient. So we need to be able to propagate this homochirality to the rest of the network, to all chiral molecules, such that we realize a homochiral chemistry. And of course, the this process needs to be persistent during the environments of early Earth until a transition to nascent biology is satisfied and everything is basically all set. So this is the schematics of the problem. And through my PhD research, I basically worked on different aspects of this puzzle. And in my talk today, I'll demonstrate a mechanism for breaking the symmetry and then a simultaneous amplification mechanism to reach homochirality at a compound that is essential to prebiotic chemistry. And then I'll talk about a system where the chirality established at this compound can be propagated to the rest of the network. So the symmetry breaking mechanism is coming from an effect called chiral-induced spin selectivity, which was discovered in 1999, and its discovery was recognized, I guess, better in 2011. And the person who discovered this effect is Ron Naumann, and jointly with David Waldeck, and he's actually listening to us in Zoom right now. And I will describe the first seminal experiment, which basically explains what this effect is about. So the researchers basically took a gold surface and put a homochiral layer of double-stranded DNA molecules, which are right-handed molecules. And then they shine light on the surface and they eject electrons from the surface. And of course, gold is not a ferromagnetic material, so the electrons that are ejected are basically represented in equal amounts of upspin and downspin electrons. And then they basically probe the spin polarization properties of these electrons after they go through the chiral monolayer. And very interestingly and unexpectedly, they find a massive spin polarization that is reaching up to 60% at room temperature. And this experiment has been replicated several times and also for other molecules such as left-handed proteins in addition to right-handed double-stranded DNA. And what they observe is the spin polarization direction for, let's say, the left-handed molecules are flipped when you flip the chirality. So essentially, this effect basically establishes a strong coupling between electron spin and molecular chirality that hasn't been envisioned before at room temperature under ambient condition. And it's a very, very strong effect. And we will utilize this effect to address the chirality, homochirality problem of the origin of life. And before going there, I will also talk about how this effect also manifests itself in a different system. So essentially, because chiral molecules strongly couple to electron spin and filter out electrons based on their spin alignment, they essentially can be thought as if you polarize chiral molecules, meaning if you push charge from one direction to the other, essentially you do this and that get accompanied by spin polarization. Because, right, the charge transport inside the chiral potential is spin selective. So essentially, if you have a left-handed and right-handed chiral molecule that are represented by these helices, and if you charge polarize this, meaning if you push charge of electrons from one side to the other, that charge polarization gets accompanied by a spin polarization, meaning a charge polarized chiral molecule becomes transiently magnetic. The theoretical understanding of this effect is still, there's no like a comprehensive theory to describe this effect, and I'm happy to answer questions if you're curious about what can be the contributing physics to this effect, but that is today the beyond the scope of my talk. So how did we utilize this effect in our research? So if you look at it, naively, it seems like it's not what we exactly want to do, right? Because what the researchers did is they basically utilize chiral molecules as spin filters. So they utilize them to select one spin over the other. But we kind of want to do the opposite thing, right? We want to pick one chirality And we can utilize this effect in basically in the other direction, meaning using a bias in spin, we can select one chiral form over the other. So that's essentially what we're going to do. And I'll talk about now the experiments related to that. So how will that work, right? Because that's probably like not obvious when you look at it at a first glance. So first of all, you need a bias in spin. And an easy and environmentally plausible way to obtain this is if you use magnetized surfaces that are abundant on early Earth, such as the iron oxides, and they give you an alignment in their spin. And essentially, if you think of a surface process on these spin-polarized surfaces, imagine like two chiral molecules, right, that are in equal amounts approaching this surface. So as they do so, because of van der Waals forces, they get inductively charged polarized, and which are represented by these sigma plus and sigma minuses. And that charge polarization is agnostic to chirality. It's the same for left and right-handed molecules. But when you charge polarize these molecules, just as we discussed before, this charge polarization gets accompanied by spin polarization. So essentially, these charge poles now become also spin poles. Okay, that is the critical part. And now these spin poles interact with the spin polarized surface due to spin exchange interactions. And these interactions now, let's say for the right-handed molecule, will be up-down like energy states, like a singlet. And for the left-handed one in this illustration will be up-up triplet-like interactions. And the energy difference between this is the energy difference between two, basically the spin exchange energy difference, which is much larger than the room temperature fluctuations. So essentially this system, meaning like doing a surface process on magnetic mineral surfaces with chiral molecules, has the ability to basically effectively increase the concentration of one enantiomer of a chiral molecule over the other. And this can be done at room temperatures. And it must be emphasized that this is not a magnetic field effect. Magnetic field effects are typically weak, and you need very high magnetic fields. But this is coming from spin, right? This is coming from spin exchange interactions, and they're actually very strong, on order of like several hundred milli-electron volts. So this is a robust effect at room temperature, which can break the chiral symmetry in an environmentally plausible way. So this was about the symmetry breaking, and this happens at the surface of a magnetic mineral, right? But we also talked about the amplification. So if the effect is just happening at the surface, then how are we going to attain more excess as we kind of go into the bulk of the system? How are we going to propagate this enrichment? Well, that can be done by crystallization. So chiral crystals crystallize in two forms. The more common way is so-called racemate, in which a unit is basically composed of the left and right-handed molecules in equal amounts. And the more like rare way, I guess, the 10% of all chiral molecules crystallize as a conglomerate. And in the conglomerate structure, each chunk is enantiopure. And if you start from a racemic mixture, of course, the overall mixture is homochiral, racemic. But the conglomerate crystallization, essentially, if you seed it once, basically propagates itself, because these crystals attract only one enantiomer of their kind, right? So conglomerate crystallization essentially is a great way of propagating the attained surface level enrichment at a spin polarized surface. So now this is our amplification mechanism. So you can see where it is going, right? So we will break the symmetry at the surface by crystallizing a molecule that crystallizes as a conglomerate on the surface. And then this enrichment will be propagated in the crystal structure. So essentially, we will use achiral magnetic surfaces as chiral seeds because of the CISS effect. So that's the kind of the physics part, but we also need to do this for a molecule that is relevant to origin of life, right? Because doing this on a given molecule is interesting, but it's not relevant really to like understanding the origin of homochirality. So we need to find a suitable molecule that satisfies our needs and satisfies these properties such that this mechanism that is due to the CISS effect can actually be utilized to understand the origin of homochirality. So to do that, we need to do chemistry, unfortunately. And this is the more bigger, more detailed diagram of John's chemistry. And it basically shows how to make different amino acids and nucleotides. And I would like to draw your attention to this line. And you don't really need to understand these structures, but what is happening is you start from hydrogen cyanide, you make sugars of different different amounts of carbon atoms, and then you make the first chiral sugar, and then from that you start making precursors of nucleotides, and then in a few steps of very high yielding reactions you make ribonucleotides. Okay. And there is one particular molecule that is very central to this chemistry called RAO that can be actually a very good fit for utilizing our mechanism. So let's just look at this shortened diagram because it's simpler to understand. We first make the first chiral sugar ever in this chemistry, and right after it's made, the sugar basically gets incubated with another molecule that is derived from glycolaldehyde, another precursor, another prebiotically plausible molecule, to make these family of molecules called pentose-aminooxazolines, and one of them is RAO. And RAO is a direct precursor of ribonucleotides. It is a very stable molecule in terms of its chiral properties. So once you attain this molecule in its enantiomer form, it doesn't racemize in solution, and it doesn't lose its homochirality. Moreover, it's not very soluble at all, so you can crystallize it easily from its low amounts. And very importantly, its chirality directly determines the chirality of ribonucleotides, and therefore RNA. So essentially, we took this molecule from its racemic solution and crystallized it on a magnetized surface. And this is the simple experiment that we did in the lab by essentially, as I said, starting from the racemic mixture of RAO and RAO-like molecules. And then we used a magnetite surface, which is an iron oxide, and it is the most commonly occurring and most magnetic mineral on Earth. And by magnetizing it with the magnet, we were able to obtain RAO molecules in their highly enantiomer-enriched form. So on the left, you see RAO molecules, RAO crystals on the surface, on a magnetite surface. And essentially, these crystals were obtained from a solution that contains these molecules in equal amounts. And then the symmetry gets broken by the surface due to the CISS effect. And the crystallization property of these crystals basically amplify the surface level enrichment. And when you collect these crystals and analyze their chiral properties, you basically figure out that the overall mixture of these crystals are highly enantiomer-enriched. And you can analyze them optically by a method called circular dichroism spectroscopy, by basically using the selective interaction of polarized light with these crystals. This is just the analytical method. And what you find is if there is any signal, positive or negative sign, that basically corresponds to a chiral enrichment. And by the strength of this signal, you can basically understand how much enantiomer enrichment you have. And the absence of the signal means that there is no enrichment. And for the downspin state, you basically get left-handed RAO. And for the upspin state, you get right-handed RAO. And the enrichment is an unprecedented level of enantiomer enrichment that reaches up to 60%. And this is like orders of magnitude higher for compared to the previous attempts to address homochirality by utilizing other chiral agents. And it's really robust at room temperature, and it has been done on a molecule that is central to the synthesis of RNA at the center of prebiotic chemistry. And just to point out again that this is not a magnetic field effect, we did this control experiment on a silicon surface, keeping the magnetic field present in the experiment. And because silicon surface is not magnetizable, although the magnetic field is present, there is no spin exchange energy difference that is coming from the surface, so you don't get any signal, meaning the crystals that you collect are racemic. And you can repeat this process again and essentially get homochirality in the second crystallization step on the surface. And you don't even need to start from 60% EEs. And we figured out that if you start from around 20 to 25% EEs, you can reach the homochirality, and that is already below the average enrichments we got after several trials of this experiment. So essentially, what we say is we can get homochirality, meaning 100% EE in 2 crystallization steps, which has never been achieved before, and especially under environmentally plausible conditions, and especially for a molecule that is relevant to the chemistry of origins of life. So that was the first important experiment. And then now I'll talk about another one, which I did partially in Weizmann Institute and in Jerusalem as well. And you see Yael and Deb who helped me. And I actually forgot to mention Ziwei's name, who you saw before, who provided the molecules. It's a great synthetic chemist. And now I will talk about how we can get surfaces that are spin polarized in a more enriched form under natural conditions, okay? Because the initial experiment we talked about was utilizing a spin polarized surface, but uniformly. And although you get spin polarization under natural conditions, you don't get uniform spin polarization. And now I'll talk about, again, by utilizing CISS effect, how can we get uniform spin polarized surfaces under natural conditions? And thankfully, you don't need to do anything different. Actually, you can use the same principle, and now in kind of the other direction. So as you can tell, like in my PhD, I just took something and just did the opposite thing, and then again. And here, we started from an enantiomer solution, and we started from now a racemic surface, so-called. And when I say racemic, I don't mean that there's chirality, but the spins are represented in equal amounts. Okay, the surface is completely achiral. It's just an iron oxide. So again, because as the molecule approaches the surface, it gets charge polarized, and that charge polarization gets accompanied by a spin polarization, and that spin polarization makes the molecule transiently magnetic near the surface. And as the molecule approaches the surface, that transient magnetic pole selectively interacts with the spins of the surface and magnetizes them, meaning spin polarizes them in a chirally selective way, again, due to the same spin exchange interactions. Okay? And this shows that under natural conditions, just by utilizing chiral molecules, you can spin polarize magnetic surfaces utilizing just the opposite nature of this effect. And you can actually see this by an interesting method called magneto-optical Kerr effect microscopy. And in this microscopy, you basically use polarized light to image magnetic surfaces. And as a polarized light basically gets reflected off of a surface, its polarization gets rotated due to the magnetization of the surface. And then you interfere this light with the initial one, and then any polarization contrast corresponds to the magnetization difference. So basically, the contrast in the images is because that there is a net magnetization difference between 2 areas, and then these areas are the ones that are covered by crystals and the ones that are not. So here in the optical image, you can actually see the crystals themselves, but because it's a magnetization contrast imaging, you don't see the crystals themselves, but they're in this white box. And as you can tell, as the magnetic properties of the surface are changing, the crystals are kind of pinning the magnetic domains because of the effect. And as you can even tell, the effect spreads about and beyond the area that is covered by chiral crystals because of kind of an avalanche like effect, right? Like if you magnetize domains underneath a chiral crystal, the magnetic interactions within the material themselves can kind of magnetize a nearby spin and then the nearby one. And then so this, this spin polarization can kind of propagate beyond the area that is covered by the crystals in kind of a nonlinear way, like an avalanche. And you can kind of see what it does in a more quantitative way in this Kerr hysteresis measurement, which is done on two different areas. And in one, we looked at an area that is in the nearby of the chiral crystals, but not exactly underneath. So this is the red area. And the blue area is away from the chiral crystals on the magnetic surface, okay? So a way to read this hysteresis loop is to start from here. And here you apply a strong negative magnetic field, and you basically magnetize these domains. And then in both ways, right? This is a strong magnetic field, so you get magnetization in both places. And then you start increasing the magnetic field in the opposite direction. And then as you reach about like 1.5 millitesla, the free domains basically give up and then they flip, okay? But interestingly, the ones that are around the chiral crystals do not. The chiral crystals basically effectively add another magnetic source due to spin exchange interactions, which basically makes the surface magnetically more strong. And of course, if you apply more field, then these also give up, and then you reach here. And then in the opposite direction, you get the same effect. And the added chirally induced magnetization is about 20 times stronger than the Earth's field. And it is even on an area that is really not covered by the crystals, it's just on the nearby. So essentially, this effect applied in the other direction can basically induce a persistent magnetization that is stable against external field fluctuations or thermal fluctuations. So essentially, you can change the magnetic field in the environment and the magnetization will be preserved. And it's also uniform at the surface. And what we only care about is the surface magnetization because that interacts with the chiral molecules. The bulk of the material doesn't really do that. And also this, persistent magnetization really shows us that the same chiral bias will be present in the environment in kind of a consistent manner. And you can easily tell that these effects can be utilized in conjunction with each other, right? Kind of like a feedback. Because if chiral molecules induce magnetization and magnetization can pick one chiral form, you can utilize them together like a feedback. So the first effect that I talked about was enantioselective crystallization, and the other one, the second one, is chirality-induced magnetization. And look at this feedback case, where we start from a completely racemic system, both in this spin and also in the molecules. So you have a surface, let's say, that has no magnetization, and then a solution that has no chiral bias. So stochastically, as one molecule starts crystallizing on the surface, because of effect number 2, it magnetizes the domains underneath itself. And on an area that is larger than the area that is covered by the crystals themselves. And then now these areas start attracting the right-handed enantiomer faster because that's the one that preferentially crystallizes on that spin direction because of effect number 2. So then you start covering these areas faster by the right-handed molecules, and then the effect kind of spreads around. And then it kind of like propagates in a kind of nonlinear fashion by using these effects together. And in a kind of environmental setting, you can kind of think of this together, right? You start from a racemic system. You have a slight bias in the magnetization of the sediments that is coming from Earth's field, let's say. And then that induces some enantioenrichment, not as great as we, let's say, obtain in the lab. But that enantioenrichment now can be propagated in the next crystallization step. And also now, because these are homochiral crystals and because of effect number 2, these molecules now, as they kind of grow, right, they magnetize the domains underneath them because of effect number 2 and induce a uniform surface magnetization because of that feedback effect that I talked about. And this surface will be now present persistently in the environment, which can be utilized in the subsequent crystallizations racemic incoming molecules. Okay, so these were the experiments that are related to the interaction of magnetic surfaces and chiral molecules. Now I will talk about the geochemistry aspect of this research, which is basically about how can we get these magnetic minerals on early Earth? What are their magnetic properties going to be naturally? And also how much bias we can get from the Earth's environment. And I've done these experiments partially in Cambridge in Nick Tosca's lab with Clancy, and also in Roger's lab here, and Perry helped me. So we envisioned this processes happening in kind of a shallow lake that is covered by magnetic sediments. And of course, there'll be incoming streams that are feeding this lake, just as was discovered in Gale Crater in Mars, actually. And that is actually this paper. And in Mars, they discovered these shallow lakes with actually highly magnetic rocks. And we kind of envisioned this on the magnetic part of the surface as a stream of racemic molecules flow through the system. They basically get filtered in a chirally selective way because of the interactions that I talked about. And if you can think of these kind of in a sequence of multiple lakes, you can essentially pick them in a certain area and filter one enantiomer out, and then dilute away the incoming solution, which contains probably a slight enrichment in the opposite form in this kind of connected lakes to each other. And John's chemistry anyways needs a flow setting to do basically reactions of the synthesis of different molecules that require slightly different chemical conditions. So it is a perfectly plausible and actually required environmental setting. And multiple of these environments are available on early Earth. So the first experiment we did was about measuring how much natural magnetization we can get as kind of a modal system to see, first of all, if there's any enrichment from Earth's field, just to convince ourselves, and then how much would there be. And then we went to a pond and then collected sediments. And you see here Roger Fu, who is a professor in geology. And because he's a geologist, he does the dirty work, not us. We are kind of watching here, and we are kind of coordinating him. And we collected these sediments from this lake and put them in a kind of glass and basically let them settle under Earth's field and then measure their chiral properties utilizing a technique called quantum diamond microscopy, which takes advantage of nitrogen vacancy centers in diamond, which are basically centers that can sense magnetic fields from small distances. And this is the cross section of the sediment. This is gravity up. This is gravity down. So this is the bottom of the sediment. This is the top of the sediment. And the quantum diamond microscopy measurements show that there are lots of magnetic sources in the sediment, first of all. And this is what we we didn't include any artificial enrichments. This is just a natural sediment that we collected from the lake. And then these are just the sources. And if you look at their dipolar orientations, again, using the same method, you basically take the gradient of those sources and then get a lot of magnetic inclusions and their orientations. There are two ways to understand if there's a bias. One is you just look at this for a long enough time, and if you're smart enough, you can see that they're pointing rightwards. The other way is to use Python, and I use the other way. So, and then when you analyze them using some statistical methods, we basically found that different sediments from different areas, that is under the same field, basically, because we are in the northern hemisphere, and that's a large scale magnetization. We basically found the same average spin alignment for different sediments, and then the direction basically matched the magnetic field direction of Boston. So they're consistent with each other, and they're consistent with our expectations. So that's great. And that means, I mean, although there's a small bias, there is a small bias in a natural setting, which is kind of a model system that can be utilized to understand early Earth, right? And then the next experiment that I did in Cambridge was about making these magnetic inclusions and how are they made and what are their magnetic properties under natural settings. And early Earth was very rich in iron ions, the ferrous iron so-called. And when it's oxidized by UV light, under alkaline conditions, which are actually also the conditions that is required by John's chemistry, these iron ions basically first go to a state called white rust. It's just ferrous hydroxides. And then with the aid of solar UV radiation, which is present in the environment, you can oxidize them to a metastable state called green rust, which then oxidizes to magnetite. And then these sediments basically acquire magnetization as they are small particles. And because of that, their magnetization is very strong. And then they get included in this matrix of these sediments of other minerals, right? And this is called orthogenic magnetization. And this is different than taking a slab and magnetizing it with an external magnet. This is essentially beating the demon when it's small. So you take individual magnetite crystals and basically it is very easy to flip them as they're small. And then as they settle, you magnetize them and then they basically cover the lake. And actually this is the highest magnetization source you can get in a natural setting. And it's much, much larger than, again, I said, taking a surface and applying an external field, which is kind of our intuition. We don't really have an intuition for this. And then we analyzed, first of all, we studied this chemistry. So that was interesting because this is the prebiotic way of making these inclusions. And the second thing we analyzed is we looked at the magnetic domain properties. And magnetic domains basically form because of magnetic interactions in materials. And the magnetic domain behavior is very dependent on the size. And as particles are small, they are in the so-called, very small, in the so-called superparamagnetic state. And they can basically respond to any fluctuation. So they're great for getting high magnetization, but they're very flimsy. They can flip all the time. When they're very large, the magnetic mineral basically cannot contain the magnetic energy and split out into domains, such that the overall magnetic energy is canceled by the domains of opposing magnetizations. So essentially, somewhere in between is great because it's both robustly spin-polarized and also it is highly spin-polarized. And when we analyze these inclusions, utilizing a technique that is developed by Richard Harrison of Cambridge University, he basically measured the hysteresis of these minerals, and by looking at the remnant magnetization ratio to the coercive field, you can actually find out the magnetic domain behavior, and the inclusions we made were in that middle regime, where the magnetization is strong, but also not very flimsy. So, okay, well, we are getting there. We talked about getting the molecules in a homochiral form, breaking the symmetry, amplifying it, and doing this in a natural setting on surfaces that can form in a highly spin-polarized fashion on the conditions of early Earth. But so far, we still talked about the molecule that is RAO. And as I promised in the beginning, I was going to talk about spreading homochirality that is attained at a molecule to the rest of the chemistry. And that is the final part of my talk. How do we propagate this attained homochirality at the central precursor? Sorry, I'm hungry a little. My teeth are going to be green now. Okay, so one way of doing this is going to each and every chiral molecule in John's chemistry and doing this process on every single of them. And that's not very elegant. And also, it probably wouldn't work on each and every chiral molecule because each and every chiral molecule in John's chemistry doesn't satisfy the properties that is required for this thing. A better way of doing this is utilizing the attained chirality and stereoselective interactions of chiral molecules with themselves to propagate it. So, and then if you ask the question, what would be the right way of doing it such that we can effectively propagate? Okay, so I will ask you to do something very dumb. I'll ask you to forget everything that I talked about today and just think about what would be the right molecular class to propagate homochirality from if you could somehow achieve homochirality at that molecular class. And by molecular class, I mean it can be nucleic acids, it can be amino acids and peptides, or it can be metabolites. Just forget about all the things and just don't make any assumptions about how to obtain homochirality. What would be the right way of propagating it? So it's not about the origin of homochirality, but it's about the propagation. Well, if you look at metabolic cycles, so meaning molecular class called metabolites of doing this, we actually run into a problem. So this is the Krebs cycle, and it is central for all life, and it contains some chiral molecules. And actually, you can enter this cycle through tartrate, which is a chiral compound that was studied by Louis Pasteur, and he discovered chirality with this molecule. And then by reducing this molecule to malate, you can actually enter the Krebs cycle. And then we asked the question, okay, let's say we can do this. Will we be able to propagate this attain homochirality in a metabolic cycle? It turns out that as soon as malate enters the cycle, it either oxidizes to oxaloacetate or dehydrates to fumarate, which are both achiral molecules. So essentially, you lose chirality as soon as you obtain it. Same for isocitrate. It is surrounded by achiral branch points. So essentially, without chiral enzymes that is deriving this cycle, Krebs cycle is basically a chirality grinder, right? You put chirality in, you can't get it out. You can't propagate it from or through it. So it is not a great class of molecules to start homochirality. Well, what about amino acids or peptides? There are some amino acids that can be crystallized in their conglomerate form, and there are just 3 of them, but there are 20 other amino acids, and with idiosyncratic chemistries. So, you know, they're not sharing the same chemistries. Also, these molecules are very soluble, so you can't really crystallize them unless you go to very unrealistic concentrations. Plus, going every single one of them and doing it again is going through that like tedious route, unless there's an easier way of doing it. And an easier way of doing it can be through using RNA to make peptides. And this chemistry, again, not surprisingly, was discovered by the group of Sutherland. And Longfei and Sam had done incredible work on this. And what they figured out is that if you use homochiral RNA to make peptides, you can essentially propagate the propagate homochirality using the stereoselective coupling of amino acids to tRNA analogs. Or in simpler terms, right-handed RNA makes left-handed peptides much faster. So essentially, from RNA, you can go to peptides, meaning like the ligated amino acids, in an effective and a general way. That's more important. This is actually working for multiple amino acids that they study, and it's always D2L. And later work actually studied the opposite form, meaning they used left-handed artificial RNAs, and they found that it is right-handed peptides. So essentially, this is an effective way of propagating it. So then the genetic molecule, RNA, essentially, is a great way to start homochirality because then you can propagate it to peptides. And then peptides, left-handed peptides, make left-handed enzymes. And if the enzymes now derive the catalysis, and if you kind of do the metabolic activities using these enzymes, you can now basically get these molecules in their homochiral form. Now the chirality will not be ground by the Krebs cycle. Well, then the genetic molecule seems like an ideal place to go and make the peptides homochiral and then onwards the metabolites. How do we get to, how can we make tRNA homochiral? Well, if we make the individual units homochiral, then RNA will be homochiral, and we need that anyways because, as I talked before, without making these monomers homochiral, we cannot go to an effectively replicating system. And if there was a shared precursor that can make these nucleotides homochiral, which is RAO, then you reduce the colossal problem of homochirality to making a single compound homochiral from which chirality can be propagated first to RNA, and then through steric-selective coded peptide ligation to peptides, and then through enantioselective catalysis to metabolites. And I think this is a much more robust, elegant, and a general way of solving the problem. And in fact, if you look at the direction of information flow from genetic molecule to peptides, it resembles the chiral information flow that was established by Francis Crick in the famous central dogma, which actually goes from nucleic acids to proteins, meaning it's the same exact direction. In our case, it's going from nucleic acids to peptides and then metabolites. So it is the same chiral information flow. And this was kind of interesting to realize for us that, you know, the chiral information, the binary chiral information follows the same pathway. And that addresses basically the all parts that I promised to address in my talk, and I'll end it here. And of course, I couldn't go into the details of all my work, but if you're interested, there are multiple papers that I published, and in these ones, you can see these work in detail. And thanks for listening. But I will not stop here, actually. I'll continue. So this was just science. So, well, I have more interesting things to talk about because this is great, and you know, it was good science for me, and it was very interesting, but it wouldn't be possible without the people who helped me to do it. And now, in the second part of my talk, I'll talk about these people. First of all, I would like to thank the members of Sasselov Lab, and every single of them are amazing people and really helped me to go through, you know, a lot of processes, including transitioning my PhD research group and the project. And Oren, Corina, Victor, Sarah, Gabby, Jonah, Emma, Christina, and Kelly, thank you. And also, I would like to point out that there were friends in the lab, not only colleagues, who put up with me during this process. Victor is here. He came all the way from California. And Oren was a dear friend while, and we had a lot of fun. So if you kind of put an Israeli and a Turk in the lab, I guess they fight a lot, and and we fought like basically in scientific discussions, I mean, every day of the week, and it was amazing to be with him and learn from him. And I visited him actually in Israel. You see here us with his family, and he was an incredible person, and we're still in touch. And if he's here, thank you, Oren. And Corina is our postdoc and lab manager who helped me a lot, and she's a great friend, and she's also a dancer. This was from her dance competition. And you see Kathy, who is my colleague as well. John told me once that you should pick your colleagues very wisely, especially you should work with creative people, and who is better than a child in terms of creativity. So I really love them both, and they helped me a lot. And also, I would like to thank the members of the Origins of Life Initiative, great colleagues and friends you see here, us in Western Australia, in Shark Bay, actually, in a field trip. And during my PhD, I had the privilege to work with, you know, really great people. And this is, as you can tell, a project that includes physics, chemistry, geosciences, rock magnetism, paleomagnetism, and I'm not expert in none of them. And without their help, I wouldn't be able to do basically anything. And I also had the privilege to work in different countries, different continents. I worked in Harvard, Cambridge, meaning both Cambridges, and Israel as well. And I would like to basically thank two people first, and then one afterwards again, who greatly influenced myself as a scientist and as a person. And one is John, who is here actually sitting. And he basically, I mean, discovered the chemical origins of life, you know, and he is one of the greatest prebiotic chemists alive, if not the best. And we always had pleasure while discussing science, and we had a lot of fun. So when I was going through my photos, I realized that, I don't know why, but all our photos with John involves at least a bottle of wine, or maybe two sometimes. And John is a great scientist. He has a lot of medals, but he keeps them in his drawer. And he sits in a small office, same size as his students, and he discovers the chemical origins of life from that small office. An incredible scientist, incredible natural philosopher, in my opinion, with an amazing British sense of humor. And I learned so much from him, really. Like, I learned all chemistry from him. So if I made any mistakes, it's because of my wrong understanding, not because of. And the other person is actually listening us. He is probably a future Nobel Prize winner, Ron Naumann, who discovered CISS effect. He is the godfather of the CISS effect, and he's a force of nature, really. I mean, he just is always in the excited state. He always likes to discuss science. And when I went to Israel, because of jet lag, I was waking up very early. So I was going to lab at like 5 p.m. and Ron would be there. And he would come like 5 a.m. in the morning and say, so what is happening today? What are we going to discover today? I read this paper and like, this is great. And like, sometimes we go for dinners and while everyone is drunk and, you know, kind of chit-chatting, John, like Ron comes and says, you know, I kind of found the answer to your question. Do you want me to tell you right now? And he is always like, he has this childlike curiosity and really like, his drive and creativity is like second to none. And after these discoveries, he actually visited me while I was working with John in Cambridge. And of course, I had to bring him to Eagle Pub. And Eagle Pub is the place, this historic bar in central Cambridge, where Watson and Crick celebrated the discovery of DNA helix structure. And they basically had party there for 6 days, 6 consecutive days. So I had to bring him there. And we went there while his short time here. Yeah, I hope still you're here, Ron. I really enjoyed working with you. And I mean, none of these would be possible without my advisor. And he really, really helped me. And through like all these processes, you know, I had a lot of difficulties. I changed groups. I had to learn chemistry, which is not easy. And one person really single-handedly brought me like to like life, and that's Dimitar. And he really supported me like all this process. You know, what I presented you here today is the 101st experiment after 100 failures and 100 ideas that just went to trash, and never once again Dimitar spared his support. And he always motivated me to push forward and just motivated me to be independent, think, and creatively, and just made me happy, you know. And so people really get anxious, I think, before they talk to advisors. And when I talk with Dimitar, I always felt much better. So that's really good. And he connected me with the best scientists on this planet and made me establish friendships and collaborations, and really treated me like a colleague and supported all these collaborations. And without him, these connections, you know, with some of you are here and they wouldn't happen. And also, I think he showed me an example of my future self as a mentor. Like before meeting Dimitar, I always thought that, you know, if you're very smart and like kind of good scientist, I guess you need to be arrogant and, you know, you shouldn't care people that much. Like Dimitar is just the opposite. Like he is an incredible scientist, very creative scientist, very accomplished, but his accomplishments are strongly correlated with his humbleness. And it has been an amazing time working with him, really. Yeah, that was hard, sorry. Well, I mean, we're at Harvard, so I really like to thank my amazing committee members. Misha is here. I took several classes from him. I had a lot of discussions with him. Like when I start my PhD, it was very scary actually to go to his classes, and I was always waiting for, so what is the intuition question? And he really like made me push to be a better scientist and just think true science, you know, and it's not just results or details, it's just the intuition and the creative ideas behind it. And without him, I really like wouldn't be able to do it, because he really like forced me and scared me to do it. Because if I wasn't scared of him, I wouldn't be able to really do it myself, you know. So like, thank you, Misha, a lot. And Marcus is the person who brought me to Harvard. Without him, I think I wouldn't be here. I don't know if he did a good thing or not. So that's like up to you. But I first worked with him as an undergrad, and he basically did everything he could do to bring me here as a PhD student. And he's an optics master. I learned a lot of optics and engineering skills from him. And Jack is another important person who really influenced me and motivated me, just inspired me through my PhD. And he is like critical way of looking at things always made me think twice before I talk to him. And he's extremely analytical, extremely creative, but also detail oriented. And he really pushed me to think more. And he also made my work famous, really, because he has been to Stockholm, so he has some fame. And I would never imagine to be on a Turkish newspaper with Jack. And also someone here. It's really an honor to have met him. Actually, Matt is here. So Matt Meselson, I mean, great friend, really. And yeah, I mean, Matt is the person who discovered how DNA replicates, and he also discovered mRNA. So, and he was the last student of Linus Pauling, and he gave me this book because we became friends after he came to one of my talks, and we started exchanging some ancient Greek texts, because actually Matt is a biologist, but he studied classics, because at that time you had to do that, and gave me this book with this very nice note. And also I would like to thank the Harvard physics staff, because without them, really, I mean, nothing works here. Like, they're amazing people, and they're the people who make Harvard work. So thanks. And also the CNS stuff and MR stuff, the people at the X-ray facility and the people at the CD facilities like really, really helped me. I also should thank the people of Greiner Lab, because they welcomed me to Harvard. They taught me a lot of engineering skills, without which I wouldn't be able to do what I did afterwards. Especially to Aaron Cron, who is now an optics engineer, and back then he was also an optics master. And I learned a lot of optics from him and engineering from him, and he really also was an entertainer. So before that, I worked with Jeff as well. So Jeff, Aaron, and Robert, thank you so much. My friends, the Turkish gang, I would say, they're always here to support me. They're still here in this room. Their friendship really means a lot. The other people, again, in the Harvard, MIT, you know, Turkish mafia, I would say. They entertained me a lot. My friends at Harvard Physics, people used to have Greiner Lab. You see in like 3 a.m. in the morning, we're like trying to solve some P sets. Probably Misha's class, actually, because they were really hard. You see our friends here, Masha, Pasha, Nate, everyone you see here. And Kaylee really motivated me to pursue everything good in this life. I really thank her as well, and other friends. So now, also I would like to thank my, like some special thanks maybe to my friends that I met through my life. Sometimes, some of them Harvard, some of them later. And Mithat is one of the greatest Turkish physicists, in my opinion. And he actually looks like a physicist, unlike me. You see us in the Library of Institute of Advanced Studies, because he's working there. And he really is a very humble person and helped me a lot. And David, I met him later in my PhD and became friends immediately in one of the conferences that Dimitar organized. And it's really great because I know after becoming friends with David, somewhere in the world, there's someone who would understand me and laugh at my jokes. And David also has a British sense of humor, like John himself. And he always sends me these like cartoons in the middle of the night, also because Seattle is 3 hours behind. So, and these jokes, he actually told me to put this because he thinks this is funny. So I put it, this chirality killed the catling. And Houston is probably knows me since I was a baby, and we are friends since then. This is our only photo, although we knew each other for 20 years. He never takes photos, and he has his very friended people like Neil Armstrong, and he's really into astronomy, and his meticulousness, his detail-oriented work ethic really inspired me all my life. Furkan is one of my best friends from high school. He really friendship was amazing all those years. And I should thank my jiu-jitsu friends because I learned as much skills in those mats as I learned in the classrooms, how to be tough, how to be determined, and how not to quit. And without jiu-jitsu, I would be a different person. I also would like to thank people of Dreyfus Foundation, Neil, Alex, Paige. We shot an amazing film and learned a lot from each other. And just a small ending with my family. I hope you're still here. And I started my journey in like a city, kind of like a relatively small city of Trabzon. I went high school there and then to Ankara for college. And on those years, my family supported me all the way. I would like to thank the Turkish science community and the people in my YouTube. They really supported me a lot, and they're still here. The Turkish people really supported me all these years. And Consul General is here. The former Prime Minister is listening us now over Zoom. I just never walked alone. I felt the whole country behind me, and that's an amazing feeling. I would like to thank my physics teacher in high school. If he didn't believe in me, I wouldn't pursue a career in sciences. These people are amazing motivators, and they always supported me. The people of Bilkent University, my art history teacher, my friends there, the Dean of Science, always behind me. Also my advisor. He always was a strong believer in my skills, and he just motivated me and to pursue science, and his generosity, desire to share his knowledge and support me made me a real scientist. And he is a theoretical physicist, which you can tell from the way he holds this award diploma, actually. Yeah, I mean, the first day I stepped in college, he just said, I want to be advisor of this kid. And he did, and he was my advisor all these years. One of the smartest people I know in this world. My uncle, he thinks he's the smartest person in the family. He probably is, but he has shown me all the great books when I was a kid, Feynman's and everything. Without him, it would be hard. And my greater family, my cousin, my aunt, and my dad. Dad also doesn't like to take photos, so this is our only photo with him on the right. And mom likes to take photos, and she's a badass woman. She does ice climbing, climbed Everest, and part-time she does gynecology. And my grandparents really raised me, and really, they taught me everything that I know, and because I was brought up my grandparents, so without them, I wouldn't be here. And my significant other, for being such a generous lover, I really thank physics because everything I could possibly ask for is thanks to her. And my love in this subject really brought me alive. I feel joy because of my love to this subject. But I know that she's also seeing others. Thank you. Thank you.
Great talk. And part two too. So the committee has to get together soon.
Okay.
Do we have time for a question or two?
I'll just log out from Zoom and we can call Jack maybe.
Do we have time for questions? Yeah, let's do a couple of questions. Well,
Defendant. The best defense is offense.
Yeah.
So. Here.
How long does it take to propagate?
Oh, I see. That's probably like the crystallization timescales. So that depends on concentration, pH. But I mean, in the lab, that took like a day, but because we kind of made it in a way, in a natural environment from a less diluted solution, probably would take longer, but still, you know, short compared to other timescales like geochemical and chemical timescales.
Experimental side, and the other question on the conclusion side. For the experimental side, the crystallization of the IO from the solution, it needs fairly concentrated. So I don't know how.
It actually doesn't. No, because it's very like insoluble on like alkaline pHs. Like we worked like around 20 millimolar solutions. Yeah.
But if you use a ribonucleotide instead of using IO, be likewise selected by some.
Great question. Great question. I don't think it would be because the conglomerate crystallization property is essential. And the nucleotides, for instance, do not crystallize as conglomerates. So that central precursor is really the way to do it because it both makes other nucleotides, but also crystallizes with that special property. Yeah. Thank you. Yeah.
But if there is another specific surface that would select the opposite enantiomer, they would claim that their enantiomer, which is opposite,
right,
would also propagate.
Right.
So at certain point, the two camps would duke it out.
Well, the thing is, like in a local environment, right? Like when we think of origin of life, we really think of a local thing, like not a global scale thing. And in a environment like a lake or connected ponds, the magnetization that is provided by Earth's field is really a hemisphere scale phenomenon. And that guarantees that different parts of the lake will have the same magnetization. Do you remember on the globe, I had like 3 dots? They actually are from different spots and they show the same direction. Therefore, they should select statistically the same enantiomer.
You don't think there would exist a surface with that?
Well, on average, no. Yes, and that's what the experiments suggest as well. Thank you. No. Right. So I mean, in the experiment that I showed, just for the purpose of time, I showed the nickel gold one because for Moki experiments, we needed that, but we actually did that one on magnetite as well. So in both experiments, we replicated on magnetite. Yeah. It's just like, because it's a pictorial measurement like Moki, that's why I showed that, but we optically also did that on magnetite. Yeah. Thank you. That's a great question. I didn't clarify it.
Okay, that's wonderful. Now we have an important meeting, which means that we have to kick you out. But let's thank the speaker. And just in case this is successful, which probably will be,